Okay, so on this, we have already talked about most of innate immunity. We talked about the external defenses, that if a pathogen tries to invade you, there are some things that prevent it from actually getting inside you. And then if it does get inside you, there are some generic defenses, phagocytic cells. Phagocytic cells, what do they do? Does anybody remember? Nathan? They do phagocytosis, which means they engulf the bacteria, remember? And then it fuses with a lysosome and it destroys the pathogen that way. Antimicrobial proteins, tell me what they do. Okay. Well, your skin does that. That we're thinking of as the skin. So the skin does secrete molecules that reduce the pH of the skin so that you don't have bacterial growth in terms of colonization on your skin. The antimicrobial proteins, this is what happens inside of our body. So there are molecules that are released from cells and then they do something to the bacteria. So let's say this is your bacteria. Your antimicrobial proteins, what they do is they trigger the bacteria to lyse. So it kind of like pokes holes in the bacteria and the contents of the bacteria and to spill out, and therefore the bacteria dies. That's one action of the antimicrobial proteins. The second thing is that they can act on viruses. This is talked about a molecule called interferon. And so viruses, what they do is they get into your cells. So this is your cell, so this is your human cell. They get into your cells and then they replicate and make more viruses. And then what happens is these viruses can leave and go infect another cell of yours and so on and so forth. So the antimicrobial proteins with viruses, these interferons, stop their replication from happening so that you only have this infected cell, not a bunch of infected cells, all right? And so that's how they work. And so these are more generic ways to fight against the infection. And then I think the last thing that we talked about, did we talk about natural killer cells in this class? No. All right, the last thing we talked about was the inflammatory response, which is this here. So your inflammatory response, remember, was the cells release histamine, which causes your blood vessels to dilate, more blood to flow, and then increases the leakiness of your blood vessels. So these phagocytic cells, blood clotting elements, they'll cause it to clot. Antimicrobial proteins leak out, and the antimicrobial proteins begin to act on the bacteria. The clotting causes the blood clot to form, and then your phagocytic cells then engulf all your bacteria and you go back to normal. And so because of all this stuff coming out of your blood, that's why swelling occurs in the inflammatory response, and the increased blood flow is why it appears red. And so that's where we're left off. So we're on natural killer cells, I think. This is the last part of your innate immune system.
We did do this.
We did do this? Okay.
All
All right. So, we're fine. Okay. So the natural killer cells we've already talked about in terms of that they attack infected cells. So those are a little bit different than all the other things. So we were starting. Okay. So in this class we started the acquired.
We finished this too.
Immune system. So this is where we're at.
Yep.
All right. I don't think I know I drew a picture, right?
Yeah.
All right. Let me finish talking about that here. And so I'm going to redraw it because I'm going to refer back to it and so on. So I drew here, we're talking about antigens, and the acquired immune system activate lymphocytes, which are B and T cells. And the B and T cells react against an antigen that gets into your body. And I think we talked about, if I recall, that there are free antigens, right? Did we write this down?
No.
Okay, I think, all right, so there are free antigens, and then there are antigens that are attached to pathogens. So yesterday I drew that you have these antigens attached to your pathogen. And I think, and I labeled them, right?
Yeah.
All right, so I labeled these as antigens. And then you have these spots on the protruding from the antigens that are called epitopes. And these guys can vary from antigen to antigen. So let me just talk a little bit about here the pathogen that has the antigens on the top here. It's the shape of these guys here on the epitopes, the shape of that is protruding out of there that distinguishes and helps your body to distinguish what's not part of itself. So therefore, a pathogen, let's say this is a bacteria, the shapes of those epitopes on the antigen sticking out are different than on your cells. So on your cells, you have also antigens with the shapes on them, but they're different than the pathogen. So that's how your body recognizes something that's foreign. So when something that's foreign comes in, it recognizes these shapes and says, okay, that's not part of us, so we must get rid of it, okay? And so that can be attached to a pathogen, or the antigen can be free. And so let me give you an example of what that means. The antigen in general is something that when it gets into your body, it triggers your body to launch an immune system attack. That's what an antigen is. And it doesn't necessarily have to be just a bacteria or virus. So examples of free antigens could be a toxin, could be a chemical, a molecule that has a particular shape. It could be pollen from plants. And later on, towards the end of the chapter, after we understand everything, we'll talk about the relationship of this with allergies too. We'll relate this to allergies here. And so those are examples of antigens. So antigens can be free, but the thing is that the antigens as well that are free or attached to a pathogen have specific shapes that our body recognizes as foreign and therefore elicits a response to. All right, and the shape is what's important. And so notice here that this picture shows you antibodies, and so notice the antibodies have complementary shapes to these epitopes and allow the antibodies to bind to them. And we'll learn later that this binding of antibodies helps to get rid of the antigen out of your body. All right, so helps to destroy the antigen, and so therefore helps you to get better if the antigen is causing you to be sick. So let's look at now. So this is how our body recognizes it as foreign. So then our acquired immune system elicits the response of B cells and T cells. So you get this antigen with this odd shape that's different than your shapes. And so that triggers your B and your T cells to have a reaction to that. So let's look at that. So antigen recognition by lymphocytes. So your B and T cells are the ones that recognize this foreign shape. So two main types of lymphocytes circulate in the blood of vertebrates. Because remember, this only happens in vertebrates. It doesn't happen in other animals. The B lymphocytes are called B cells, and the T lymphocytes are called T cells. A single B or T cell has about 100,000 identical antigen receptors. These antigen receptors on a single cell recognize the same epitope. Antigen receptors, these guys here, these guys can bind to the antigen. Let's look at the B cells first, and I'll show you what I mean by the identical antigen receptors and that they're all the same. So your B cells receptors bind to specific intact antigens. Secreted antibodies or immunoglobulins are structurally similar to B cell receptors, but lack transmembrane regions that anchor receptors in the plasma membrane. You have some space underneath here, so I'm gonna draw a few pictures to illustrate what I mean with B cells here. So Here's a B cell. B cells protruding off of it have these antigen receptors, and this is the shape that the antigen receptors have. It's kind of Y-shaped here. I'm going to kind of go like this. All right. And it says can have, we just wrote that it has about 100,000 identical antigen receptors. I'm definitely not going to draw 100,000 of them, but I will draw 2. So they have a whole bunch of these. But the key is all the antigen receptors look exactly the same. And the thing that is important about these antigen receptors on these B cells is this part here. I'll highlight here. So these guys are the same. So all 100,000 of them have the same shape at the end. Why is that shape important? This is the shape that can bind to an epitope. So notice here I drew here an indentation that's pointy there, and that could bind to this particular epitope. So they can match up together. So, so this is, I'm gonna write, has a complementary shape. To one epitope on an antigen. And can bind to it. So this part right here can bind to that epitope right there. That's what it means by B cell receptors, that first bullet. B cell receptors bind to specific intact antigens. So the antigen's intact, it's not broken up or anything like that. It's part on the surface of this pathogen, and those can bind to it because of that. And we have lots of different kinds of B cells. So this is one type of B cell. Another type of B cell, what makes one B cell different than the next is what type of receptor, the shape at the end. Let me ask you this, with this pathogen, could another type of B cell bind to this one? Yeah, absolutely, because look at this epitope is different. So a B cell that has the shape where it's more curved inside of here, that type of B cell could also bind to it. So therefore, different types of B cells can bind to that pathogen depending upon how many epitopes you have. The second thing, so that's the first thing that B cells do, is they bind to the pathogen. We're going to see later that that helps also to get rid of the pathogen. The second thing that B cells do is that B cells, this is number 2, they secrete antibodies. Now let me draw a picture of an antibody. Antibody. What do you notice about the shape? Exact same as the receptor. All right, exact same as the receptor, which means, and the important thing here is that this part of it that the antigen can bind to it. So guess what? These antibodies can also bind to that epitope. All right, secrete antibodies which can bind to the epitope as well. Okay. So this can come over here. We could draw another arrow to that. And both of those can help to rid the body of the pathogen. So the shape of those epitopes are really important, and the cells and the antibodies, the B cells and the antibodies, have to have a complementary shape to bind to that pathogen. All right, so let me show you what this picture is illustrating. So this picture here, the left side is talking about B cells. So they're drawing you a picture of a B cell. These little Y-shaped things sticking out are just like the little Y-shaped receptors that I just drew. They're enlarging one of them here. They're peptide chains, and it tells you light and heavy chains and things like that. You don't need to memorize all of that. You don't need to know that for this class. Just the general idea that we have a receptor that's in this Y shape, and at the end here, you have an antigen binding site, which is what I illustrated here, and so on, that the antigen can bind to. So that is B cells. So now we're going to look at the second lymphocyte, which is T cells. T cells also have receptors on them that can bind to the antigen, but they look a little bit different. So this is what the T cell looks like. They only have, when the receptor comes out, they only have one antigen binding site. So one here versus two. And so T cells also can bind to that. The difference is that there are some differences between B and T cells. So the T cells receptor consists of two different polypeptide chains. That's what the two purple lines are. It's just making one receptor. That's what you need to know. All right, so what happens is the T cells, and how they're different than B cells. T cells bind to antigen fragments that are bound to cell surface proteins called MHC molecules. MHC molecules are so named because they are encoded for by a family of genes called the major histocompatibility complex. That's where the MHC comes from. So what happens, let me draw a little cell off to the side here. So here's a cell, here's the nucleus, and so this is one of our cells. We have DNA in the nucleus. The DNA, section of the DNA is a gene. Genes code for making proteins. So this DNA codes for making these MHC molecules. So we have this I'm just going to put a circle, it's not a circle, it's a square. MHC molecules. And so we have these, and so then what happens is if this cell, so a normal cell, this is what a normal cell looks like. If the cell becomes infected, so let's say, this is really large compared to that cell, but let's say it gets infected by this pathogen. The pathogen has these antigens, on the outside. What happens during infection is that the antigen, pieces of the antigen, or they call antigen fragments, float around in the cytoplasm. So these fragments of the antigen, I'm going to draw here. So I'll draw the triangle here and a half circle here. So these guys are antigen fragments. And this, we would say, this is an infected cell. So what happens is that when the cell becomes infected, and now it has these fragments of the pieces of antigens here, the MHC molecules that we make, what they do is they pick up these fragments. So I'm going to draw here a little half circle on the end. And guess what they do? They migrate to the cell membrane, like this, and present the fragment to the outside of the cell. That's what infected cells do. So now, this human cell that has been infected has really like an epitope of the pathogen. Does anybody have an idea of why they do that? Why would you do that?
So a B cell could attach onto it and break it down.
That's right. It's a way to tell, hey, this is an infected cell. We need to do something with this. We don't want to keep infected cells around, all right? So we need to do something with that. Because if you have, this is an infected cell and a non-infected cell doesn't have that on the top on it. And so therefore, the B cells can recognize that, hey, we can bind to this and this cell has been infected. So it's a way to tell infected versus not infected cells. So let's write here what I just drew here. This is what this says. So infected cells produce MHC molecules which bind to the antigen fragments, are transported to the cell surface, and present that antigen. It's called antigen presentation. So what happens then is that the T cells can detect that antigen fragment displayed on the cell surface. Now it says depending on their source, these antigens are handled by different classes of MHC molecules. The classes of MHC molecules, I'm not gonna we're gonna talk about two classes, class 1 and class 2. You don't have to memorize them, you just need to know the general differences between them. And so, so let's look at that. So these class 1 MHC molecules are found on almost all nucleated cells of the body, like red blood cells don't have a nucleus. They display peptide antigens to cytotoxic T cells. So let's look at this picture here. Let's look at the left side, because this is what we've just talked about here. So here's your infected cell. So notice here are your antigen fragments. So there are two different kinds of antigen fragments from the infection. And then notice here, this green thing, this is your MHC molecule. So I'm just gonna write, they put it in two different parts here, but it's all collectively MHC. All right, so that's your MHC molecule. So notice here the MHC molecule has, notice here this one has a specific shape to fit into and present that antigen fragment. So therefore it migrates to the membrane, as we said before, and prevents that antigen. So the class 1 MHC molecule, I'm gonna write presents the antigen fragment. And that's the antigen fragment right there. And that's how we know the cell is infected. So what comes next is the cytotoxic T cell. Cytotoxic T cell has also a receptor. So this is the two parts of this receptor. The T cell receptor binds to the antigen fragment. And that activates the cytotoxic T cell. What do we say? What do we want? What do you think this infected cell, what do we want to do to it? Do you think? We want to destroy it, all right? We're going to kill this. And so that's what the cytotoxic T cells are involved in, is killing these cells. So we'll talk more later on in the chapter about how exactly the cytotoxic T cells destroy this cell, but they do destroy this cell. So we're going to kill off this infected cell. And so, so that's one side of this. The other side of this picture here has a different, starts with a different cell. Notice here it says antigen presenting cell. And so I'm going to give you examples of what kinds of cells those could be. These could be macrophages. And I'm going to introduce a new type of cell, dendritic cells. What they have in common is that they both are phagocytic cells. They both take in microbes through phagocytosis, kind of like what we talked about yesterday. So that's what we have here. We have this cell. This cell is a natural part of our immune system, and it's doing us a favor by taking in the microbe and going to destroy the microbe for us. So this is not an infected cell. This is a cell doing its job to kill off the pathogen. But what happens in that process of taking in the microbe is that it's kind of similar to what happens in the infected cell, in that the antigen fragments float around in the cytoplasm. So what that allows is, again, your MHC molecule, so this is your MHC, to pick up those fragments and display them. So we have the same thing going on here that we do in the last picture. So these guys are displaying here. So this is displaying the antigen again. But instead of the cytotoxic T cells binding to it, we have a different kind of T cell called the helper T cell. So these guys, we don't the helper T cells work differently than the cytotoxic T cells. These guys destroy the cell it binds to. Why is that a good thing? Because it's an infected cell. But notice this cell, this isn't infected. This cell's just doing its job. It's destroying your microbes. So we want to help this, all right? So a helper T cell, we're gonna see when the helper T cells are activated, they help other cells of the immune system to help with the attack against this pathogen. So they help other cells. So then to describe what part A here and part B, we could say in this part here, the presentation of the antigen fragment by what kind of cell in A? By what kind of cell? An infected cell. It's by an infected cell. Activating what kind of cell at the end? Activating the cytotoxic T cells. Cytotoxic T cells. The difference between that and this, we can write the same sentence, we're just going to change the presentation and what we're going to activate. So here the presentation of the fragment is by, well they're both displaying. Yeah, the antigen presenting cell. So that could be a macrophage, this dendritic cell. This is any type of phagocytic cell that's going to take in the microbe by phagocytosis. So this is the presentation of the antigen fragment by, I'm going to write, I guess I can write antigen presenting. Sometimes they abbreviate these cells, APC, antigen presenting cell, activating a helper T cell. So they have different end results. So the difference between B cells and T cells, let's look at the difference. B cells bind directly to the antigen. So the B cell receptors can bind to it, or the antibodies they secrete bind directly to the pathogen, the antigens. These guys, T cells, don't bind to the intact antigens. They don't bind to free-floating antigens or to the antigens on your pathogen. T cells only bind to cells that have either been infected or taken in the pathogen. All right? And so that's a big difference between B and T cells. Okay? So that's a general overview of B and T cells and what they do. So we're going to take a look a little bit more in depth here. Oh, and these two bullets are about the second picture there. So class 2 MHC molecules are mainly on those dendritic cells, the macrophages. They display antigens to the helper T cells. So that was the second part of that picture. So B and T cells. Let's look at some differences and a little bit more in depth about what they do. First of all, their development. Lymphocytes, B and T cells, arise from stem cells in your bone marrow. And these newly formed lymphocytes are alike, but later on develop into B or T cells depending upon where they mature. Stem cells. Anybody know what stem cells are? Okay.
Are they just pretty much cells that don't have a specific action that can be done? So like I don't know the scientific
It's okay.
part of it, but that's just what I know.
So they don't have a specific action, like a job.
Yeah.
All right. Want to add to that?
I've heard that a lot of stem cell research is involved programming the stem cells to reproduce, I guess, and create new tissues. So there are cells that can be programmed to do specific actions.
right. So they are, at first, the stem cells, they don't have a specific job, but can divide and become different types of cells that do have specific jobs. And we'll learn later on in the course, there's different types of stem cells. There's embryonic stem cells, so early on in development, you know, when you're first developing, you have to be able, your cells have to develop into eye cells and cells of your stomach and skin cells and so on, and have special jobs. Your stem cells in your bone marrow can't become any type of cell in your body. They're different than, but they can, there are some stem cells in your bone marrow that are lymphoid stem cells that can become different lymphocytes, so B cells and T cells. So what happens is these stem cells in the bone marrow, early on in development, they're very similar, but then eventually they, what we call differentiate or become specialized into their specialized functions, into B or T cells. And the location where that occurs differs. So if we look at this illustration here, the rectangle on the left represents, that's inside your bone marrow. And so you can see your stem cell, the lymphoid stem cell, is inside the bone marrow here. So right on the top here, there's the stem cell. If the cell differentiates and continues in development within the bone marrow, it will develop into a B cell. So B cells develop in the bone marrow. The T cells finish their development, they move from the bone marrow to the thymus and finish their development in the thymus and then become a T cell. So these guys now are differentiated. A B cell has a different job than a T cell. So they're specialized. Both of those can now enter the fluids in your blood, the lymph in your lymphatic system, and so on, and do its job. So the B cells, the job is to bind to antigens and secrete antibodies that can bind to antigens. The T cells can bind to infected cells or cells that have engulfed a bacteria. All right, and so once you develop these B and T cells, the next process is the removal of self-reactive lymphocytes. So as B and T cells mature in the bone and thymus, their antigen receptors are tested for self-reactivity. Lymphocytes with receptors for antigens that are already in the body are destroyed by apoptosis. So what does this mean? When you make your B and your T cells, and therefore the receptors, you make all different kinds of them, thousands and thousands of them. The difference between them is the shape on the end of the antigen receptors here. And what happens is sometimes you can make B and T cells that the shape will fit into the antigen on your body cells. So let's say your body cell, so this is a body cell of you, and you have an antigen, let's say, that has this shape here. So we can actually make B cells. If your body makes a B cell that has the antigen receptor that's complementary to that, so it has it's really big, but like this, it can bind to that and attack your own body cells. It can make antibodies against your own body cells. We don't want that to happen. So therefore, in development, this is where it says tested for self-reactivity. That's what that means, is this B cell and T cells that we make, is it going to attack our own body cells? And if it does, we don't want those, so we destroy them. All right, so they're destroyed by apoptosis. And therefore, you only have B and T cells that have antigen receptors for antigens that you don't have. All right, and so then that's why you know for sure that when they attack the B and the T cells, that it's just something that's foreign to you. If this doesn't happen to you, what happens? You don't. That's how autoimmune disorders happen, all right? So that you don't kill off those B and T cells, and so you do get destruction of your own tissues, all right? So I'm gonna write here, failure to do this equals autoimmune disorder. So that can lead to an autoimmune disorder. Okay, so let's look at now, what actually happens, what do the B cells and the T cells, we're just gonna look at the B cells probably today, to do to, when you get an infection. So we're gonna look specifically here. So in a primary immune response, binding of an antigen to a mature lymphocyte induces the lymphocyte's proliferation and differentiation. This is called clonal selection. Clonal selection of B cells generates a clone of short-lived activated effector cells and a clone of long-lived memory cells. We talk about that first bullet in a primary immune response. Primary means first. So this means that this is the first time your body has ever come into contact with a particular kind of bacteria. So the bacteria gets into you for the very first time in your life. That's what that means. And so it says binding of antigen to the mature lymphocytes, the B cell, induces the lymphocyte's proliferation and differentiation. Proliferation means it divides. And differentiation means it specializes. It's called clonal selection. So I want to illustrate this process with the next picture here, and I'll talk about why it's called clonal selection. So here you have a picture of your B cells. This is at the very beginning. So when you make all your B cells in your bone marrow, you have thousands of different kinds. They're only showing you 3 here. What's the difference between these 3? The receptors, right? So this one has sticking out here, it's kind of pointed. This one is the opposite, it's kind of indented, pointed, and then this is a square. So what's different between all your B cells is those receptors. You have thousands of them, but you don't have very many of them. You only have a few of each kind. And so what happens is when you first get infected, here's your antigen molecule. So you first get infected, what your body has to do, here's this foreign molecule. Your body has to figure out which one of these B cells is going to work against that antigen, because you have thousands of different kinds. And so that's what this is. This is the selection part of clonal selection. We have to select which B cell is going to work against this antigen. And so in this particular picture, it's the middle one, because the middle one has the complementary shape. This one's more diamond shaped, and so it can fit right in there. So the antigen molecule can fit into those receptors. So that's the B cell that we want. So here we have, I'm going to write up here for this first thing here. I'm going to put selection of B cell with the complementary shape to the antigen. So this is the one. That takes some time. So then what happens? From that cell in the middle to the next picture, what does it look like happens? It divides and makes lots of new ones. All right? So these guys divides, and makes clones, clones of the B cell with the complementary shape. So we get lots of those B cells. We don't divide and clone any of the other ones, only the ones that have the complementary shape to the antigen that we're getting infected with. Question?
So is this where acquired immunity comes in? Where
Yes.
the B cells, like for example, when you get a flu shot, it's basically dead flu cells, I guess? And your body just gets used to the...
Does it get used to it? Give me a second here. Like,
Okay.
all right, give me a second here. And it has to do with these guys. This part right here. All right, so hold on for a second. All right, so therefore we get lots of these different B cells that have this antigen, complementary antigen binding site there. So then what happens is some of these cells, so some differentiate in order to specialize into, I'm going to put here, memory cells. And then the other, some differentiate into plasma cells. So, let's look at plasma cells first. What does it look like the plasma cells are doing? Yeah, they are giving off antibodies. Those are those free molecules that look like the receptors there. They have the similar shape here. So when it says antibodies, I'm going to add the word in front of it on the top of here. They're secreting. They secrete antibodies. And remember, antibodies have the complementary shape that these antibodies can go and bind with the antigen molecule. All right, and we're going to see later on that that helps to get rid of the antigen. So these guys are launching an attack by secreting antibodies. These guys are short-lived cells. So they only live long enough to secrete antibodies to fight your current infection. And then once your infection, once the antigen is gone, your antibody concentration goes down and these guys die off and your infection is gone. So these are short-lived cells. And so these are the cells that are helping you with your current infection. These cells have nothing to do with your current infection. So these guys are working against the antigens that you're currently infected with. These memory cells, now you have a bunch of these memory cells, and these are, let's write here, these guys are long-lived. So these guys can stick around sometimes for a lifetime. And what happens is you've got this army of these memory cells, and what happens the next time you come into contact with the same antigen, so it gets into your body a second time, you have this whole army of memory cells that immediately start dividing and releasing antibodies and start attacking your antigen just like that, very, very quickly. You don't have to do this whole process. The first time, your body has to figure out, okay, which B cell is going to work, then make more of the B cells, then differentiate into the plasma cells, which will then release the antibodies that will help you to fight the infection. So oftentimes the first time you come into contact with either the bacteria or the virus, you get sick because all that takes a while to happen. But the second time you come into contact with that, then it reacts a lot quicker. You've got all these cells already produced and ready to launch your attack. And so therefore the second infection is much quicker. And this, going back to Joe's question about vaccines, this is how vaccines work. So vaccines, when you get a vaccination against the flu, let's say, so you get a flu shot, what they will do is take the most popular strains of the flu virus from the previous year. So which ones were most people got sick by. And they'll take those and they give you a weakened or dead, or they take out the genetic material of the flu virus and leave the shell, so the antigens are still present, and they give it to you, and then so your body reacts to those antigens and goes through this process and makes memory cells against those strains of flu, so that when you come into contact with in real life that same flu virus, it gets into your body, so you breathe it in or something like that, it has the same antigens, you've already got memory cells that will then immediately start producing antibodies that will fight the infection, and therefore you don't get sick that way. The thing with the flu shot and with that vaccine, though, is that it's a guess. They take the most popular strains from last year, and that doesn't necessarily mean that that's going to be what's prevalent now. And so when people say, oh, the flu shot doesn't work, you can't control, what happens is the flu virus mutates very quickly. And so therefore, if it mutates and has different antigens, your flu shot's